Welcome to the ultimate microscopic metropolis, this cell. In this explainer, we're shrinking way down for a grand tour of the bustling factory that forms the foundation of, well, every living thing. Think of it as a highly advanced, non-stop city where millions of tiny workers are coordinating every single second just to keep you alive.
But first, let's rewind. Did life really kick off in boiling hot springs? Scientists from the Birbal Sani Institute were studying the Puga Valley hot springs in India, and they found conditions mirroring early Earth from about 3.5 billion years ago. They actually discovered that calcium carbonate deposits in these extreme, heat-loving environments might have shielded the very first organic molecules from harsh radiation. That protective shield could have helped form the very first cell membranes. It's just wild to think that the ancestors of our microscopic factories were born in such a hostile, boiling environment. Here is our tour map for today. 1. Origins and cell theory. 2. The basic cell structure. 3. Osmosis and cell transport. 4. Meet the organelle workforce. 5. Plant versus animal cells. And 6. Cell division and death. Section 1. Origins and cell theory. How we found the cell. You know, before microscopes, we literally had no idea this hidden world even existed. The foundational cell theory actually took nearly 200 years to unify all of biology. It started in 1665 when Robert Hooke looked at a thin slice of cork through a microscope he designed himself. He saw these tiny box-like compartments and decided to call them cells. But it took decades before Matthias Schleiden and Theodor Schwann put the pieces together in the 1830s, realizing that all plants and all animals, every single living thing, are made entirely of these cells. Then, in 1855, Rudolf Virchow dropped the final puzzle piece, stating that new cells arise only from pre-existing cells. Think about what that actually means. Every single cell in your body right now came from a pre-existing cell, forming an unbroken chain extending all the way back to those ancient, boiling hot springs. Section 2. The basic cell structure, city borders. So, every city needs a border wall, right? For a cell, that's the plasma membrane, and it's what we call selectively permeable. It operates on a fluid mosaic model. Imagine a fluid-moving sea of fats, or lipids, studded with a mosaic of proteins acting as specialized security gates. Now, this membrane is incredibly thin, only about 7 to 10 nanometers thick, yet it's the ultimate gatekeeper, strictly controlling what enters and exits based exactly on what the cell needs at that very moment. Now, plant cells, they're packing an extra layer of armor, a rigid outer cell wall made mostly of cellulose. This is absolutely vital because plants can't just get up and walk away to find shelter. They have to withstand the elements right where they are. This leads to a fascinating phenomenon called plasmolysis. When a plant cell loses water, the flexible cell membrane inside actually shrinks and pulls away, but that rigid outer wall stays perfectly intact. It maintains the overall shape of the box, which is just a brilliant structural defense. Section 3. Osmosis and cell transport. Moving resources. Let's talk logistics, specifically how resources cross those borders. Enter osmosis. Osmosis is the movement of water from an area where it's highly concentrated to where it's less concentrated, right through that selectively permeable membrane. If you put a cell in an isotonic solution where everything is balanced out, nothing changes. But check out hypotonic and hypertonic conditions. This explains a classic science experiment. Drop a piece of potato in pure water, a hypotonic environment, and water rushes into the potato cells, making them swell up so the potato gets nice and crunchy. But if you drop that same potato into heavily salted water, a hypertonic environment, water rushes out of the cells to dilute the salt, leaving the potato totally rubbery and limp. Section 4. Meet the organelle workforce inside the city. All right, welcome to the factory floor. First up is the nucleus, the grand control center wrapped in a double-layered membrane. It holds your DNA, which are literally the master blueprints of your entire body. Then you've got ribosomes, these tiny structures that read those blueprints to synthesize proteins. But doing all this heavy lifting requires power. That's where the mitochondria step in. They handle cellular respiration, breaking down glucose to pump out ATP, the energy currency of the cell. But here is the most mind-blowing part. Mitochondria actually have their very own DNA and ribosomes completely separate from the nucleus. Because of this, scientists are pretty sure they used to be free-living bacteria that permanently moved into our cells billions of years ago. Next up, logistics and storage. The endoplasmic reticulum, or ER, is your massive manufacturing network. The rough ER is studded with ribosomes churning out proteins, while the smooth ER synthesizes lipids and hormones. Once those goods are made, they head over to the Golgi apparatus. Fun fact, Camillo Golgi first spotted the cellular post office in an owl's nerve cells back in 1898, and people honestly didn't believe him for a long time because it was so hard to see. But he was totally right. The Golgi modifies, packages, and ships vital products in little vesicles to wherever they're needed. And finally, we have vacuoles, serving as the storage tanks to keep water, minerals, and waste safely contained.
And we absolutely cannot ignore the lysosomes. Discovered by Nobel laureate Christian Dadov, these act as the factory's waste disposal system. But they have a much darker nickname, the suicidal bags of the cell. Why? Well, they're packed with up to 40 types of incredibly potent digestive enzymes. Mostly, they clean up debris and destroy invading bacteria. But if a cell gets severely damaged, these bags intentionally burst open, releasing those enzymes to digest the entire cell from the inside out. It's a programmed self-destruct sequence called autolysis, an extreme but totally necessary sacrifice to keep the rest of the organism healthy.
Section 5. Plant the animal cells, rival microscopic cities. When we put these two side by side, it's pretty clear how different lifestyles demand completely different architecture. A stationary plant cell leans heavily on a rigid outer cell wall to brace against the wind and rain. It's got chloroplasts to cook up its own food from sunlight, and one massive central vacuole to hold water and maintain pressure. If that vacuole empties out, the whole plant wilts. An animal cell, on the other hand, is all about flexibility and movement. It skips the cell wall entirely, relying on a flexible membrane, and uses prominent lysosomes to quickly digest complex foods. Let's zoom in on the plant's specialized gear known as plastids. You know the reason a leaf is green, a flower is bright red, or a potato is packed with starch? It all comes down to these plant-only organelles. Chloroplasts hold the green chlorophyll needed for photosynthesis. Chromoplasts hold vibrant yellow, orange, and red pigments, which is basically an evolutionary advertising strategy to attract pollinators like bees and birds. And leucoplasts? Those are the hidden, colorless vaults storing massive reserves of starch and oils. Section 6. Cell division and death, replication and demise. So if new cells only come from pre-existing ones, they've got to divide, right? There are two main ways this goes down. Mitosis is your standard division. One parent cell splits into two identical daughter cells with the exact same DNA. This is why the cells healing a scrape on your knee are perfect clones. But meiosis is a special two-step division used strictly for reproduction, resulting in 4 diverse cells with only half the normal chromosomes. When 2 of those cells fuse, the original number is restored. It's a brilliant mechanism that ensures you might look like your parents, but you aren't an exact copy, creating the genetic diversity that is absolutely essential for life. But this division isn't just a free-for-all, it is highly regulated. In normal animal cells, there's a rule called contact inhibition. Basically, when cells grow and bump into their neighbors, they communicate and tell each other, okay, stop dividing. It's a flawless system for keeping tissue size in check. However, if cells lose this crucial ability to communicate, if there's a genetic error and they fail to trigger those lysosome, suicidal bags we talked about, they'll just ignore their neighbors. They grow out of control, piling on top of each other, and unfortunately, this is exactly how tumors and cancers form. Which brings us to our final thought. The human body replaces hundreds of billions of cells every single day. About 1% of your entire body is completely regenerated daily through this flawless microscopic coordination. Take a second to think about the sheer scale of that. You have an invisible, perfectly orchestrated metropolis keeping you alive right now, repairing your skin, fighting off bacteria, and generating your energy. How many billions of factories are replicating inside you right at this very moment? It really gives you a whole new appreciation for the complex, beautiful building blocks of your own life, doesn't it?